Hello class, we want to resume our lecture on secondary active transport. So, a non-spontaneous transport process can also take place through coupling to another active transport process known as secondary active transport. So we have a primary active transport and a secondary active transport. So let's see what happens in secondary active transport. Okay, so primary active transport. With primary active transport, we transport, this is an antipode direction or antiparallel. Potassium is coming into the cell and sodium is going outside the cell under the expenditure of ATP. So this mechanism really defines the sodium-potassium ATPase mechanism. So let's see. We have the sodium, potassium, ATPase. It's an enzyme which pumps three sodium ions out of cells and two potassium ions into a cell or into the cell through active transport mechanism into the cell through active transport mechanism. Okay, so we have sodium, potassium, ATP. So if this is the cell, this is the nucleus, these are the cytoplasm. So as potassium goes inside the cell, two potassium ions go inside the cell. Two potassium ions, as two potassium ions go inside the cell, two sodium ions, three sodium ions come out. One may ask why? Because if sodium is pumped inside the cell, rather, there will be what? Anywhere sodium goes, water follows. So if sodium enters the cell, there will be a condition called edema or edema. The cell will be edematous. The cell will swell. It's the same as swelling. The cell will swell. Or it will be what? If it is in plant cell, it will be what? Turgid. Because what? Salt will always attract water. So if salt were to be here and potassium coming out, then salt will attract water so that water will fill the whole cell and interstitial fluid, causing edema. So potassium comes in so that it will make us very strong and active. But if potassium goes out of the cell, sodium comes in, then the person is about to die. High serum potassium is called hyperkalemia. And it is not good. People who are having hyperkalemia cannot even get up from bed. They are paralyzed. Because so potassium will activate the muscles and other things. but low serum or plasma or blood potassium level is called hypokalemia. This one too is not good. It's not good. They are both not good. So it has to be what? Normal kalemia or normal potassium level. The same way, high serum or plasma sodium level is called hypernatremia. Hypernatremia. The same way, low level is called hyponatremia. Sorry, hyponatremia. So you can see, and this is mediated by the sodium potassium ATPase. It's also called sodium potassium pump, or sodium potassium ATPase is also called sodium potassium ATPase. ATPase means that it works by using energy because against concentration gradients. Some call it sodium potassium pump. They don't even bring the ATPase. They just say sodium potassium pump. Sodium potassium pump. Some say sodium potassium pump instead of sodium potassium ATPase pump. Right, it's the same. It's a pump, and it's an enzyme. Sodium potassium ATPase pump or enzyme. Okay, so we can see there is energy because it's an active transport. So the enzyme will transport potassium into the cell and send sodium out. Because like I said, if sodium comes into the cell, there will be turgidity or there will be edema, because sodium, wherever sodium goes or wherever salt goes, water will follow. So the cell will swell up with water. So we don't want it. So that's why the sodium potassium pump pumps sodium out of the cell against a concentration gradient, meaning that the sodium in the cell is very small and the sodium outside the cell is very, there are many, highly concentrated, but still the enzyme will pump the small sodium in the cell because we don't want to entertain a lot of salt in the cell. Because if you entertain a lot of salt in the cell and other places, it leads to what? Hypertension, edema and hypertension and the rest. That's why if you are having hypertension, they say don't take in salt, because too much of salt will lead to what? Hypertension. Accumulation of what? Water in blood vessels and other things. Okay, so for the secondary active transport, this is a simple mechanism. Here we have a simple transport system. So when we are transporting something, we just add something also flows down its concentration gradient. For instance, for the secondary active transport, if this is a cell membrane, and we are transporting this and this. This is, let's say, magnesium 2 plus. It will come through through active transport, primary active transport. primary active transport. For the secondary active transport, meaning that if you are transporting this magnesium Mg2 plus, then maybe amino acid, one amino acid like lysine will also follow. It's like a concerned person. For a concerned person, if you haven't even something that does not even concern him, he will go and involve himself. So that is secondary active transport. Primary active transport, you are just transporting something against a concentration gradient. But for the secondary active transport, this thing you are transporting, there is somebody else who is a guluma, or somebody else who is a gossip, an amino acid or a protein or anything at all, will say, hey, if you are going, then give me lift, give me lift. So it will also come and join. It won't free lift. So let's see, secondary active transport. So if you have, for secondary active transport, if you have this going to the cell, going through the cell membrane, it will come here. This is primary active transport. For secondary active transport, we have this, and this thing going, is traversing the cell membrane to come here. But this one will say, hey, if you are going, I want a lift, so give me a lift. So this is what maybe benzene will say, or glucose will say, give me a lift. Glucose will say, give me a lift. So glucose will join. And down is concentration gradient. So that is secondary active transport. This is secondary active transport. By primary active transport, you transport against concentration gradient. So more of the things are here, more are here, more are here, and you are transporting. It's like what? The bad wife and the husband. She has plenty money, but she wants more. And so the husband has to work so hard to use ATP to be able to provide the needs of the family. But for secondary active transport, this one is working hard to use ATP to drive its reaction, like here. So we have more of this rectangle here, secondary active transport. More. So a bad wife, a good husband, bad wife, small salary, more salary. He says, still, I need all your money. So the good man will give all the money. But when he's giving, there is some concerned person over here say, hey, me, I will not give. I will not give. You give. I will come and eat free of charge. Then glucose will join. And it will not pay anything. It will not use anything. This man has paid everything. So it will just join. That is what we call secondary active transport. So you can see sodium is coming, then glucose will join. They are not going to the same place circle. This one is going here, this one is going here. But because somebody has hired a Mahama Kandu to go to a town, or a taxi to go to town, he said, give me lift, then you join. It will not pay, glucose will not pay anything. And that is what we call secondary active transport. And it uses same port mechanism. But the primary active transport uses anti-port mechanism. Importance of transporter systems. They help cells regulate their volume, internal pH value, and ionic environment. They also concentrate metabolites, exclude toxic substances, establish ion gradients required for sedative phosphorylation, and stimulation of muscle and nerve cells. The meaning of this is that the cell will permit some molecules or ions to pass through and some not to pass through. And this will help the cell to keep what? Homeostasis, or constant internal environment, constant volume, constant internal pH, and constant ionic environment. They will also concentrate metabolites that the cell needs to use for its own physiological activities. It will exclude toxic substances. So if there is any toxic substance like ammonia and the rest, they will be excreted from the cell and establish ion gradient. Examples of transporters, we have the glucose transporters. Aquaporin, which transports water. Glucose transporters transport glucose. ATPase pump transports what? Sodium, potassium. Sodium will go out of the cell because if it comes into the cell, it will cause the cell to swell and become edematous, hypertension. But potassium will come into the cell because if the potassium is inside the cell, it will energize the cell will be very strong and the person will not be paralyzed. Plasma membrane calcium 2+ ATPase. So we have different types of ATPase. We have the calcium 2+ ATPase, sodium potassium 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 ATPase. Just remember the charge. Then sarcoplasmic reticulum calcium 2+ ATPase, proton transporter for ATP synthesis. Okay. Mitochondria are known as the powerhouses of the cell. They are organelles that act like a digestive system, which takes in nutrients, breaks them down, and creates energy-rich molecules for the cell. So wherever mitochondria is, there is the release of energy. The biochemical processes of a cell are known as cellular respiration. So this is the structure of the mitochondria. The mitochondria has granules. It also has ribosomes. The folding of the mitochondria is called what? These folds, the folding is like this. The folding of the mitochondria is called what? The folded sections are called crista. So plural crista. And the plural is what? Cristae. Singular is crista. And plural is cristae. So this is the folding. So the folds are called cristae. They are plural, different folds. We have the intermembrane space. There's a space here. This space is called intermembrane space. Then this is the ATP synthase particles. ATP synthase is an enzyme. That's the enzyme particles are here. The matrix is the cytoplasm of what? The mitochondria. Whenever you see matrix of the mitochondria, that's the cytoplasm of the mitochondria. And this is the DNA of the mitochondria. So the mitochondria itself is what? Intracellular organelle. It is a powerhouse. So the mitochondria, more importantly, it has an outer membrane and an inner membrane separated by intermembrane space. This is the intermembrane space. This is another space, intermembrane space. Let's continue. So metabolic reaction, mitochondria is for respiration or breakdown of energy, breakdown of glucose, fat, and others to release energy. So it meets the energy demands of the body or of the cell. Takes place in energy needs using oxygen. Oxygen, aerobic respiration takes place in the mitochondria. Its significance is that it converts the energy stored in macronutrients to adenosine triphosphate, the universal energy donor in the cell. So this is glycolysis. Glycolysis, what is glycolysis? Is the breakdown of glucose into two pyruvates or two units called pyruvate, generating two ATP molecules. 2 NADH and 2 water molecules. But glycolysis alone cannot meet the energy demands of the body. So we require the Krebs cycle also and the oxidative phosphorylation or the electron transport chain. So we'll look at glycolysis in a glance.
Professor Dave, let's talk about glycolysis.
He knows a lot about the science stuff. Professor Dave explains.
Talk about glycolysis.
He knows a lot about the science stuff. Professor Dave explains.
We need energy to do literally anything, from running a race to simply breathing. And every cell in your body is furiously producing all this energy all the time. Where does this energy come from? Well, technically, it arrives from the sun. The sun is releasing enormous amounts of energy as a byproduct of nuclear fusion reactions. And that energy makes its way to Earth where it can be absorbed by plants. Through photosynthesis, which we will discuss later, plants use sunlight as well as carbon dioxide and water to make glucose. And it is all of this glucose, among other biomolecules, that becomes the starting material for metabolic processes in our bodies. This degradation of biomolecules to generate energy that cells can use is called cellular respiration, or sometimes more specifically, aerobic respiration. Let's see how this works, looking at glucose as the substrate. Aerobic respiration requires oxygen, so any organism that breathes in oxygen from the atmosphere is doing so in order to facilitate aerobic respiration. Glucose, which we can either consume as starch or break off from glycogen stored in the cell, can be converted through metabolic pathways in the presence of oxygen into carbon dioxide, which we breathe out, water, which is most of what we are, and energy, the energy we need to think and move. This is not unlike the combustion reactions that happen in an engine, so it's a reasonable analogy to view biological organisms as machines. The electron exchanges that occur throughout these metabolic pathways utilize the electron carrier NAD+ in its other form, NADH. This is a dinucleotide with an interesting base, nicotinamide, that can exist either as NAD+ with a positively charged nitrogen atom, or, if reduced, it can become NADH. This transfer, facilitated by an enzyme called dehydrogenase, helps catalyze the breakdown of glucose. Cellular respiration happens over three major pathways. There's glycolysis, the citric acid cycle, and oxidative phosphorylation. Let's focus on these one at a time. Glycolysis comes first, and it happens in the cytoplasm of the cell. This is the process by which glucose molecules are split into two pieces called pyruvate. This first pathway is actually anaerobic, meaning it does not require oxygen, so it is the most evolutionarily ancient metabolic pathway, occurring in even the simplest cells. In this pathway, one glucose molecule can yield a net of 2 ATPs. It requires 10 enzymes to happen, which catalyze each of the 10 steps, as well as an investment of 2 ATP molecules in the preparatory phase to get 4 ATPs back over several steps in the payoff phase. The names and details of each individual reaction may not be of interest to every viewer, but in case they do interest you, here is a brief overview of each step. First, the hexokinase reaction. The enzyme hexokinase phosphorylates the oxygen on carbon 6 to make glucose 6-phosphate. The polar phosphate group traps the molecule inside the cell and also reduces the concentration of regular glucose inside the cell, which encourages more glucose to enter by diffusion. This step costs 1 ATP, which provides the necessary phosphate group for the reaction. Next, glucose 6-phosphate isomerizes to become fructose 6-phosphate, a process which is catalyzed by phosphoglucose isomerase. After that is another phosphorylation, this time on the carbon 1 hydroxyl, gives us fructose 1,6-bisphosphate. This step is catalyzed by phosphofructokinase 1, and it will cost another ATP. Now, this molecule is ready to be cleaved into two smaller ones. Fructose bisphosphate aldolase is a lyase enzyme that will split fructose 1,6-bisphosphate into a molecule of glyceraldehyde 3-phosphate, or GADP, and a molecule of dihydroxyacetone phosphate, or DHAP. The DHAP will be converted into another molecule of GADP by the enzyme triose phosphate isomerase, which leaves us with two molecules of GADP. That's the end of the 5-step preparatory phase, with 2 ATPs spent to achieve the 2 phosphorylations. Now it's time for the payoff phase. Let's just look at one of our 2 GADP molecules from the preparatory phase, and we see that the first thing that will happen is an oxidation to become 1,3-bisphosphoglycerate. This requires NAD+ and a free phosphate, or inorganic phosphate, to occur, and the enzyme involved is called glyceraldehyde phosphate dehydrogenase. Next, a phosphoglycerate kinase will catalyze transfer of a phosphate group to ADP to become 3-phosphoglycerate, producing 1 ATP in the process. Since each of the 2 GADP molecules will make 1 ATP, that's a total of 2 ATPs for half the total payoff of glycolysis. Then, phosphoglycerate mutase transfers the remaining phosphate from this hydroxyl to the next one over to make 2-phosphoglycerate. Then, enolase catalyzes a dehydration, resulting in the loss of this hydroxyl group, which will produce phosphoenol pyruvate. And lastly, the remaining phosphate group is transferred to an ADP by pyruvate kinase, generating another ATP and the pyruvate we discussed before. So altogether, it's a 10-step process. The first 5 steps comprise the preparatory phase, which take 1 molecule of glucose and produce 2 molecules of GADP. This will cost 2 ATP. Then, the other 5 steps make up the payoff phase, in which each molecule of GADP will be converted into pyruvate, producing 2 ATP each in the process, for a total of 4, meaning the net energy production from 1 molecule of glucose is 2 ATP. If you are required to memorize basic facts about glycolysis, this should probably suffice. But if you wish to memorize this process in more detail, here is a table that lists the necessary enzymes for each step, as well as any relevant input or output besides the actual substrate. Certainly, the main thing to remember is that in glycolysis, glucose in the cytoplasm of the cell is converted into pyruvate, which will then move on to the next stage of cellular respiration. Thanks for watching, guys. Subscribe to my channel for more tutorials, and as always, feel free to email me, Professor David.
Let's go through a summary of glycolysis. So in glycolysis, we have glucose being broken down to 3 carbon units, and each one is called a pyruvate. This is the same as this. This is the end product of glucose catabolism or breakdown through glycolysis. Whenever you hear lysis or lysis, lysis is what? Breakdown. So glycolysis is what? Breakdown of glucose. Glyco is what? Glucose. So glucose breakdown, and lysis is what? Breakdown. It's called breakdown. Break down. So breakdown, and I said glucose is what? Glyco is glucose. Glycoprotein. Anytime you hear glyco, it's glucose. Or sugar. So glucose breakdown is called glycolysis. So we end up producing what? Pyruvate. And there are 2 units of pyruvate, 2 molecules of pyruvate from 1 glucose molecule. have pyruvate. 2 pyruvate. So let's go to the steps. So glycolysis consists of 10 steps. We split them into 3 phases. Phase 1 consists of step 1 to 3. Phase 2, step 4 to 5. Phase 3, step 6 to 10. So this is step 1 to 3. We have the glucose. Then hexokinase will add phosphate group to carbon 6. This carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. So hexokinase. A kinase is an enzyme that adds what? A phosphate group to a substrate. A kinase adds phosphate group, adds phosphate group to a substrate, adds phosphate group to a substrate. And a phosphatase, removes phosphate group from a substrate. Removes phosphate group from a substrate. So hexokinase is 1. It will add phosphate. That's the first enzyme is hexokinase. It will add phosphate group to this glucose. Then it will go to isomerization. Then it will become fructose. Then fructokinase will also add another phosphate group to carbon 1, making fructose 1,6-bisphosphate. Then this fructose 1,6-bisphosphate will be splitted into what? Dihydrogen acetone phosphate and glycerol phosphate, which will now be interconverted to glycerol phosphate, which will now be channeled through these various steps to produce our end product, 3-carbon pyruvate. Pyruvate is a fatty acid. It has only 3 carbons. So pyruvate is containing what? A carbonyl group. So it's a ketone, and at the same time it also has what? A fatty acid group or carboxylic acid group. So let's see. So these are the steps. So we have the regulatory step. The regulatory step is the same as what? Okay, so let's see. So the regulatory step is what? A committed step. So it has 3 regulatory steps. 1, 2, 3. So when I say a committed step of a pathway or of an enzyme catalyzed reaction is a step when committed step is a step in enzyme catalyzed pathway in which when that step is able to go through, then there will be a formation of what? Products. But if the enzyme catalyzing that step is inhibited, it means that products will not be formed. So that is the committed step or the enzyme or the rate limiting step. Committed step or rate limiting step. So let's go through. So we have hexokinase is a committed step. Phosphoglucoisomerase, glucose isomerase is a committed step. Then phosphoglycerate kinase step is also a committed step. Okay, so we see glucose, we add phosphate to it. We get the phosphate from ATP. Add it to carbon 6, giving us glucose 6-phosphate. on carbon 6. Phosphoglucose isomerase isomerizes the glucose 6-phosphate to fructose 6-phosphate. You remember isomerization, that's change of positions of some of the atoms or substituents on the molecule to create fructose 6-phosphate. We get phosphate group from what? ATP. And ADP is because when we break one of the phosphate group from ATP, we release ADP, adenosine diphosphate, instead of triphosphate. So the phosphate will go and join to carbon 1 to produce fructose 1,6-bisphosphate. These are all investment phase because we have invested 2 ATPs and we have used 2 ATPs, so we've lost 2 ATP energy to produce fructose 1,6-bisphosphate. And fructose 1,6-bisphosphate is isomerized or broken down to what? Dihydroxyacetone phosphate and glycerol phosphate. And an enzyme here called glycerol phosphate dehydrogenase converts all the dihydroxyacetone phosphate to what? And the glycerol phosphate using this coenzyme NAD plus a phosphate to produce what? 1,3-bisphosphoglycerate. Then ATP is produced. ADP went inside, then we took this phosphate group and added it to ADP to produce ATP. So now we have produced 1 ATP from 1,3-bisphosphoglycerate. This is 1. There are 2. We've used 1 to produce ATP. So we have 3 phosphoglycerate. Phosphoglycerate mutase will mutate it, cause a change in the molecular arrangement to produce, will bring the phosphate group to carbon 2. The phosphate group is on carbon 3, it will send it to carbon 2, making 2 phosphoglycerate, carbon 2. Then it moves it from carbon 3 to carbon 2. Then enolase will convert or add a double bond to the 2 phosphoglycerate to produce phosphoenol pyruvate. You see the double bond is between carbon 2 and carbon 3. Then ATP will also be produced again. ADP will go inside to produce, to remove this phosphate, add it to this ADP to produce ATP. Then we have our end product, pyruvate. Meaning that we produce 2 ATPs over here. ATP, the investment phase, ATP went inside and we release ADP, meaning that we have used ATP. Here too we use ATP. We use ATP here too. But when we came here, just one, the 3 carbon compounds are 2. Dihydroxyacetone phosphate and glycerol phosphate. We use one of them to produce 1 ATP and another ATP. Then the second one will also come and produce 1 ATP and another ATP. So in the end, we have 4 ATP being produced. We use 2 ATPs to produce 4 ATPs. That is because we produce 2 units of the pyruvate. And pyruvate, we are only seeing 1 here, but it's supposed to be 2. Because this is for 1, then this one also comes. So we are producing 4 ATPs. So 2 ATP going inside and to produce 4 ATP. So the net energy produced is what? 4 minus 2 is equal to. So glycolysis gives us 2 ATPs. Only 2 ATPs. It's not enough. So we have to go through the citric acid cycle to generate more energy so that we can run away from anaerobics. So general reactions involving glycolysis, glucose plus 2 ATP produces 4 ADP plus 2 PI producing, so let's say glucose plus 2 ADP plus 2 PI plus 2 NAD. We produce 2 pyruvate. Then we produce also 2 ATPs. Then we produce 2 NADH, which will be used in Krebs cycle to generate more energy. Then we produce what? 2 molecules of water and 2 protons. That's the next reaction of glycolysis. Let's look at the Krebs cycle. in more detail. Let's look at the Krebs cycle in more details. Krebs cycle is also called the tricarboxylic acid cycle.
Professor Dave again, let's talk about the citric acid cycle.
He knows a lot about the science stuff. Professor Dave explains.
We learned about glycolysis, which is an anaerobic process, meaning it does not require oxygen to occur. Since oxygen is not necessary, the first simple organisms on Earth were able to generate energy through glycolysis for millions of years. But 2 ATPs per glucose just isn't that much. In order for higher organisms like animals to evolve that can run and jump and swim, they had to evolve additional metabolic pathways that generate far more energy than glycolysis. This became possible once plants covered the Earth, thereby filling the atmosphere with oxygen, which is a product of photosynthesis. Thus, the possibility for large-scale, oxygen-dependent aerobic respiration was born. The location in the cell where this process occurs is the mitochondria, which are eukaryotic cell organelles. According to endosymbiotic theory, mitochondria seem to have been entirely separate organisms that were incorporated into eukaryotes, specifically for the respiratory abilities they possess. This activity begins with the pyruvate molecules that were generated in the cytoplasm during glycolysis. These pyruvates will enter the mitochondrial matrix to find coenzyme A. In the presence of NAD+, pyruvate will undergo decarboxylation, oxidation by NAD+, and then attachment to coenzyme A, generating acetyl-CoA. Acetyl-CoA will then enter the citric acid cycle, also known as the Krebs cycle, or the tricarboxylic acid cycle. This is an 8-step pathway requiring 8 separate enzymes. In the first step, the enzyme citrate synthase removes the acetyl group and tacks it onto oxaloacetate to form citrate. Next, with help from aconitase, a water molecule is removed and another one is added to generate a structural isomer of citrate called isocitrate. Then, catalyzed by isocitrate dehydrogenase, isocitrate is oxidized by NAD+, and then decarboxylates to form alpha-ketoglutamate. Next, another CO2 is lost, and further oxidation by NAD+ takes place with the help of ketoglutamate dehydrogenase. The resulting molecule will join with coenzyme A once again to form succinyl-CoA. CoA is then displaced by a phosphate group to form succinate, which is catalyzed by succinyl-CoA synthetase. This will make one molecule of guanosine triphosphate, or GTP, in the process, which can be used to make 1 ATP. Then, with the help of succinate dehydrogenase, succinate is oxidized by a different molecule, FAD, which will result in fumarate and FADH2. Next, fumarase will catalyze hydration, which results in malate, and lastly, one more oxidation by NAD+ takes place with the help of malate dehydrogenase to give oxaloacetate, which will restart the cycle, reacting with a new acetyl-CoA. Overall, for every acetyl-CoA that enters, this cycle will produce 3 NADHs, 1 FADH2, and 1 ATP. Since 1 glucose will produce 2 pyruvates in glycolysis, and therefore 2 acetyl-CoAs, we can double these numbers to get the amounts per glucose molecule. Once again, for the ease of memorization, here is a list of each step of the citric acid cycle with the names of the respective enzymes. We can see that there still hasn't been a huge payoff in terms of energy, but the products of the citric acid cycle will then move on to oxidative phosphorylation, which will generate the majority of the ATP produced in aerobic respiration. Thanks for watching, guys.